Alrighty, let's get started. So this lesson is lesson 2 of chapter 4, and this lesson is going to answer a lot of those questions you had when we started this whole genetics thing, like how on earth do a string of A's, T's, C's, and G's give me my curly hair or my blonde hair or my green eyes and blue eyes and dimples? Or what the heck does this have to do with my hitchhiker's thumb and widow's peak? Well, the answer is proteins. Proteins do much of the chemical work inside cells, and so they largely determine how those traits are expressed. The things that you see, the blue eyes, the brown eyes, the hair that's curly or straight or thick or thin, dimples, no dimples, all of that is determined by the existence of proteins. And the lesson we're going to kind of dive into this week is called protein synthesis. And synthesis means putting things together. And what proteins are strings of amino acids linked together. And during this lesson, we're going to learn how that whole process takes place and what that has to do with DNA. So let's start with how does a cell make a protein? So here we are during what I called before protein synthesis. Okay, cells use the genetic information on the DNA to make proteins. So we talked last week about what does that mean? Well, genes, segments of DNA bases, nucleotide bases, are considered a gene, and genes code for different traits. And we said genes could be a couple of hundred base pairs long or 20,000 base pairs long, but a section of A's, T's, C's, and G's, a section of adenines, thymines, cytosines, and guanines make up a very specific code for a trait. And what that means is they make up a very specific code for a protein that will kind of cause your cells to then exhibit the trait. So proteins, I guess you can say proteins help determine the size, the shape, the color, and all other traits by triggering certain cell processes. Okay, and I guess you could say proteins are also a way that the DNA sort of shows your traits. Now there are a lot of different players in how to make a protein. And trust me when I say we'll go through a lot of this week and learn this and manipulate this in a lot of different ways. But we need to go back and recall what a ribosome is. And I think you remember from a couple of chapters ago that a ribosome is an organelle that synthesizes proteins. Synthesize proteins. Okay, so that means that the ribosome is the organelle that puts together the amino acids to make the proteins. Okay, sorry I had to write that real fast. I'm using a new stylus tonight, which is not so good. And synthesize, well, we'll get to that. So other players are going to be a molecule called RNA. Now, RNA is ribonucleic acid. Chemically, it's different than DNA. Structurally, it is a single strand instead of a double helix. And it has adenine, guanine, cytosine, but no thymine. So no thymine. Instead, it has uracil. Okay? So single strand is a very important biological molecule if we want to get from DNA to a protein. RNA is a key factor in making this happen. There are two types, and we'll talk about them. There's mRNA, which is the M stands for messenger RNA, and there's tRNA, which is transfer RNA. And as we're moving through the process, we'll talk about those a little more. All right, so we're going to start at stage 1. Okay, the first thing we have here is something called mRNA. So that's that single strand. Remember, no thymine, has uracil. So the cell needs to make a protein. And what it does is it unzips between the base pairs. That means it unwinds its double helix and exposes a segment of base pairs. It exposes a gene, okay? And that gene is going to make a protein. Okay? So DNA is a very fragile molecule. It has to stay inside the nucleus. So it is the job of mRNA to make a copy of the gene so that it can then leave the nucleus and the DNA remains unharmed. So what the DNA does is it unwinds, as you can see in this image right here, and it exposes a set of bases, and then the RNA base pairs line up with the DNA base pairs. Okay? Again, cytosine always pairs with guanine, and uracil will pair with adenine. So however many base pairs this gene is, RNA will come in and make a copy of it. It's like the photocopier. It unzips, boom. We need to make this protein. The DNA unzips, exposes this gene, mRNA comes in and makes the copy. Okay. Then the mRNA leaves the nucleus and enters the cytoplasm. That way the DNA stays unharmed. Okay. So step 1, DNA unzips, exposes a set of base pairs for a gene, mRNA comes in, makes a copy, and then leaves and enters the cytoplasm. Once it's in the cytoplasm, it sort of moves around until it attaches to a ribosome. Now when we were doing our cell analogies, we sort of saw that ribosomes were everywhere in the nucleus. They were in the cytoplasm. They were either free-floating in the cytoplasm or attached to the endoplasmic reticulum. So this particular mRNA strand leaves the nucleus, floats around in the cytoplasm, and attaches to a ribosome. On the ribosome, the mRNA has the code for the protein. See, in stage 1, it went in and it made a copy. It takes the copy out and attaches to the ribosome. It's the ribosome's job to read the code and make the protein. Okay? So the mRNA provides the code for the protein that will be made. Okay? Now, there's another molecule here that we need to talk about because floating around in the cytoplasm are specific amino acids that are attached to a molecule called tRNA, which is transfer RNA. So the mRNA attaches to the ribosome, and the ribosome begins to move down the mRNA reading the code. It reads the code 3 base pairs at a time. Okay? 3 base pairs at a time. Okay. Next, as the mRNA gets read, 3 base pairs get read, then the tRNA brings in the corresponding base pairs. So if you look here, this first one right here, the mRNA is A, adenine, C, cytosine, A, adenine. tRNA brings in the corresponding base pairs, which would be A matches up with uracil, C matches up with guanine, A matches up with uracil. Boom. Okay. Then it moves on to the next one. Okay. When it gets to, now the ribosome keeps on moving down, 3 base pairs at a time. It gets to the next one. The next one is C, U, U. And what the tRNA brings in is G, A, A. And then it moves down to the next 3. And this process keeps going on until it gets to the end of the gene, okay, which can be 20,000 base pairs long. And as you see up here, the tRNA is bringing in amino acids. Okay? And as those amino acids are being brought in, the ribosomes are hooking them together to make a protein. Now, here we go. So the next step, the amino acids bond together, okay, and the ribosome continues to read the code. So you'll see here the next 3 bases are C, U, G, and the tRNA will bring in, CUG will bring in G, A, C. Okay. and that this amino acid will then bond with this one, and our protein chain begins to grow. The order of the amino acids is determined by the order of the codes on mRNA, which is determined by the gene in DNA. So stage 1, DNA unwinds, exposes the gene. mRNA comes in and copies the gene, then leaves the nucleus and goes to a ribosome, and then the ribosomes begin to read the code and bring in the amino acids to make the protein. So the gene is what coded for the protein that the ribosomes are eventually making. And finally, the ribosome continues to move along the mRNA, and amino acids are added one at a time. The protein continues to grow until it reaches a code to stop. When it reaches a code to stop, the protein is released. Tomorrow we'll talk about what that stop code is, because it's the same for all, which I think is amazing, but I don't want to give too much information today. I know this was a lot to take in, and we are going to review it in a lot of different ways and manipulate it in a lot of different ways. But to review, the key to protein synthesis is this: to answer this question, how do all of these nitrogen bases reflect the traits in my body? And the answer is proteins. And then how does the DNA control that? Well, the DNA controls that through this process called protein synthesis. And protein synthesis is getting the code off of the DNA, out of the nucleus, and actually made so that the body can use it. Okay, so I know that was a lot. Kind of take a look at your notes. We'll do some activities tomorrow, and have a good night.